Hello, my name is Saskia and I'm one of the creators of coX. coX is a tool that helps you to distinguish between genes that are likely to be involved in the brain disorder of your interest and genes that are not. To this end, coX uses a guilt by association prioritization approach as well as different visualization techniques in up to 7 large datasets of gene expression in the post-mortem human brain of different ages. Briefly, the principle of guilt by association says that a gene genuinely involved in a disease is likely to be part of the same regulatory network as other genes already known to be involved in the same disease. Thus, such a gene should co-express with known disease genes. In order to see how this works in practice, let's look at the example of febrile seizures. Febrile seizures are the most common type of seizures occurring in children between the ages of 6 months and 5 years, in combination with increased body temperature. So far, 10 genes have been securely implicated in this neurological disease. Nevertheless, these genes only allow for an incomplete picture of the disease mechanism. In particular, because of their role in brain function, researchers have long been wondering whether any of the 22 zinc transporter genes are involved. We are now going to use coX to find out for which zinc transporter genes, if any, co-expression in the human brain supports a role in febrile seizures. When you first open coX, you find yourself on the front page of coX, which gives you an overview of the tool's functionality. coX works in a stepwise manner from choosing datasets to finding genes, to cleaning, to prioritizing, and finally to visualizing. The first step is to use the navigation bar to head to the first step. The first step requires you to select the datasets that you wish to work with. Since the onset of febrile seizures is between 6 months and 5 years, I'm only going to select datasets that include individuals in this age range. After selection, I press the Submit button at the bottom of the page and wait until the tool is finished. Next, use the navigation bar again to navigate to the next step, where you upload the gene lists pertaining to your disease. Here I upload a list of 10 known febrile seizure genes. Then I upload a list of the 22 zinc transporter genes. Finally, I also upload a list of genes known to be involved in epilepsy. These genes are not used during the prioritization step, but including them ensures that during the cleaning stage, these genes are not used as negative control genes. I will explain what negative control genes are in more detail during the next step. Then again, press the Submit button, and the tool outputs a list of genes that it cannot find in each of the selected datasets. So again, use the navigation bar to get to the next step. In this step, the data is cleaned. You can either choose to clean the data in a conventional manner, using a combination of background correction and quantile normalization, or you can choose the adaptive cleaning procedure, removal of unwanted variation. This method uses negative control genes that are known to contain no biological variability of interest in order to estimate the systematic noise and to remove it. Usually, housekeeping genes are used for this purpose, but you can also decide to use empirically chosen negative control genes. Note that any genes uploaded in the previous steps will be excluded from being negative control genes. This might take a while, but you can follow the tool's progress at the bottom of the screen. So the next step is candidate gene prioritization. First, the tool prompts you to pick the brain developmental periods of interest with regards to your disease. So with febrile seizures, we know that the onset is typically between 5 months and 6 years, which actually corresponds to the periods of late infancy and early childhood. So don't worry if you are not familiar with this terminology. There is a handy table on the front page that helps you to convert ages into developmental periods. After selection, press the Prioritize button. Once the tool is finished, which may take some time, you can determine the tool's output. For example, you can select a threshold which allows you to control the maximum number of falsely prioritized genes. Note that this is kind of the worst case scenario. So a threshold of 20% actually only rarely means that indeed 20% of the prioritized genes are wrong. So after pressing the Submit button, the tool provides a table of all prioritized genes as well as the datasets that they were prioritized in. Note that when we adjust the threshold to, let's say, 10%, a lot less genes are being prioritized. Remember that prioritizing a candidate gene does not mean that it is genuinely involved in the disease of your interest. Prioritization only tells you which genes you should further investigate. BrainCoX includes several interactive visualizations that allow you to investigate the role of such a gene in the overall disease network and how its interactions with known disease genes change throughout development. However, note that this cannot replace functional validation as a proof of true causality. At best, these visualizations help you to rule out genes that were falsely prioritized, as well as give you a better understanding of the disease gene network. So, since the prioritization step is somewhat of a black box, we are providing a visualization that allows you to see the interactions that contribute to a candidate gene being prioritized. Let's take the genes prioritized at a 10% threshold. These are currently saved since this was the last prioritization output we generated. So in order to see what networks they form with the known disease genes, I check all the data sets and reducing the candidates to the candidates prioritized at 10%. I also changed the default from circular to free in order to get a better impression of the topological structure of the network. I then press submit again. As you can see, this renders a plot of the network. This reveals that unlike SLC30A9 and SLC30A7, both SLC30A10 and SLC30A3 are interacting with known disease genes. This makes them more interesting candidate genes. Often there is more information in the changes gene interactions experience throughout development. So to utilize this information, we created yet another visualization. If you input a candidate gene of interest like SLC30A3, the tool highlights the interactions with each known disease gene throughout the entire brain development. For example, the interaction between SLC30A3 and GABRD seems to start out as an activating relationship in early development and then changes to being inhibiting in later development. So one could actually speculate that in febrile seizures, this process might be disrupted. I hope this short tutorial was helpful for you. You can find more information on how to use BrainCoX in its documentation and also on the front page of the application. There are many more features in BrainCoX that I hope you will explore at some point. Thank you for your attention and good luck with your gene discovery.